so glad to be here, so honored to be here. Although in Texas it's about 88 degrees, so I was a little annoyed today when I ran around and felt like 30 to me with rain. But I am honored to be here, and when Gail invited me to speak on stem cell research, I said, well, I know the issues, but can't you find anyone better? And she said, no, it's stem cell research made easy. And I thought, well, if I can understand stem cell research, anyone else can. So hopefully this will be made easy. In Texas, we have been able to bring both attention and positive change to the abortion issue, but often overlooked in our movement is the issue of stem cell research, even by many who call themselves pro-life. With technology advancing unfettered, our fight for the right to life will soon be consumed by defending the lives of those in the concentration camp and the lives manufactured in laboratories. You can see these scientists here looking at embryos through their microscope, and they say the little embryos are holding their signs. Scientists are designing and selling the perfected human life, and many are hungry to buy. The science can be intimidating, thus many hesitate to speak or engage on behalf of our embryonic brethren. Many pro-life activists and politicians would rather fight the clear-cut abortion issue and leave the stem cell fight aside because on the surface, stem cell research appears to affect a broader cross-section of society. This is understandable. We are referring to tiny, microscopic, unseeable humans. When have we heard that before? In laboratories, rather than very public abortion mills. But make no mistake, we stand today where we stood with abortion in the 1970s. And embryonic stem cell research is as much an affront to the sanctity of life as is abortion. Are we talking about science and research, or are we talking about the destruction of innocent human life? These are the only two sides to the issue. You cannot be pro-embryonic stem cell research and pro-life, much to the vexation and confusion of many. When I field media calls on the matter, I always ask the reporter these questions because the reporters themselves are maybe willfully, maybe not, but they generally are confused on the issue, and they will not distinguish between embryonic stem cell research and the other types of research. When you look in the headlines on USA Today about stem cell research, it's always stem cell research. We never see the distinction of embryonic versus the other kinds. So I always ask reporters, are embryos human? If not, what are they? Is killing innocent humans wrong? If not, why? If yes, then at what point does killing become wrong if not at the earliest stages of human life? As pro-lifers, we only oppose the stem cell research that destroys human life. Stem cells serve as a repair system for the body. When a stem cell divides, each new cell has the potential to either remain a stem cell or become another type of cell with a more specialized function, such as a muscle, a nerve cell, a red blood cell, even a brain cell. Stem cells differ from other kinds of cells in the bodies. They have 4 properties. These are very general properties, but this is what distinguishes them from the other cells in the body. They're capable of dividing and renewing themselves for long periods. They can replenish and repair other cells. In college, we always hear, how many brain cells you kill when you're drinking. So I think there are a lot of college kids who are hoping that the stem cells in the brain that science will soon be developed enough to help them. They can morph, stem cells can morph into specialized cell types, also known as differentiation, and they can transform into cell types of completely different tissue, which is absolutely miraculous, and this is known as plasticity. There are 5 basic types of stem cells. There are fetal stem cells, embryonic stem cells, which will be most of the focus today or in this session. There are cloned embryonic stem cells, and there is a difference between the two, and we'll cover that. Adult stem cells, and the new technology has yielded induced pluripotent stem cells, or iPSCs. Fetal stem cells are extracted from an aborted or miscarried baby through a tissue or blood sample. The blood sample is then treated with growth factors and injected into the body of a patient. Embryonic stem cells, the second type. They are derived from human embryos the embryos are made for infertile couples and in vitro fertilization clinics. Embryonic stem cells are typically extracted after the frozen embryo, after the frozen embryo is thawed, then it's allowed to develop to 4 or 5 days old, and then the cells are taken from the inner mass of the embryo. And extracting the cells from the inner mass of the embryo does in fact kill that embryonic human. These stem cells are used to start stem cell lines. You've heard about stem cell lines, and there was a big, when President Bush, I think it was 2002, he made a decision about stem cell lines. And so that's a buzzword, and scientists are saying we need more stem cell lines, and we haven't used the ones that are already existing, and President Obama just authorized more money for more stem cell lines. So what happens is we take these embryonic stem cells and we develop cell lines. It's just a technical term to say that we use research, and it's a strain of a stem cell on which you do a certain type of research. And then this strain of embryonic stem cell research is for another type of research. Cloned human embryonic stem cells, the third type, are derived from a cloned human embryo. Somatic cell nuclear transfer is the scientific process by which a human or an animal is cloned. That's what the doctors used or the scientists used for Dolly the sheep. And those are a little bit different than the embryos manufactured for in vitro. This is fertilization, whether it's fertilization in the lab or the old-fashioned fertilization, versus cloning. And somatic cell nuclear transfer is the process by which we clone. Somatic cell nuclear transfer, can't say that very fast, involves a donor egg, so an egg, and you take out the nucleus, and then you have a little cell donut or egg donut. What happens is a somatic cell, which is basically any cell on the body, skin, hair, that somatic cell is then put into our little donut egg, and they're fused. So that egg, the donor egg of the person being cloned, the donor egg now has a new nucleus from the person being cloned. And that begins to divide, and then we now have a cloned human embryo. This is the identical method. There was a Cece the cat in cattle. This process is used to clone cattle, especially in Texas. And we're okay with cloning animals, but we don't clone humans. This is maybe a clearer diagram of, maybe it's not so clear, of somatic cell nuclear transfer, and you can see how we reach the same end. And what happens here is this is when the stem cells are taken out, and we have embryonic stem cells from the clone. When the cloned human embryos are 5 to 7 days old, that's when the stem cells are ripe enough to be extracted. And when they're extracted, our cloned human brother or sister dies. The fourth type of stem cells are adult stem cells. These are much easier to explain. And they reside within human tissue and are readily available. Now note, the word adult refers to the age of the stem cell. It does not refer to the age of the person from whom the cell comes. So when you hear umbilical cord stem cells, those are in fact adult stem cells because the word adult refers to the age of the cell, not the person. Adult stem cells are found in the tissues of umbilical cord blood after live birth, spinal cords, fat, bone marrow, dental pulp, nasal cavity in the brain, peripheral blood, blood vessels, skeletal muscle, skin, cornea. You can even take stem cells right out of your eye. The digestive tract, the retina, the liver, and the pancreas. Adult stem cells are removed from a patient in a tissue or blood sample without harm to that person. A dozen adult stem cells can multiply into millions of new cells that are genetically identical to the original extracted cells. And then the most virulent cells are put back into the patient, and they differentiate into the appropriate cell type. In human clinical trials, these new adult stem cells, they go right to the area of damage. It's absolutely fascinating. But the studies have shown that the new stem cells, they run through the body, and they find the part that needs fixing, and they get to work. And this list shows the source of the cells and what the adult stem cells, how they are specialized, what the adult stem cells are specialized for. The fifth type is induced pluripotent stem cells. You can't say that one very fast either. This one, too, is a type of adult stem cell research, but it's new technology, and the science and application are far more complex than adult stem cells, so we consider it a separate category. What happens is you take an ordinary somatic cell, like a skin cell or a hair cell or a digestive tract cell. They're extracted just from a blood or tissue sample, and the scientists are able to manipulate those pluripotent somatic cells they're able to induce them much like you would reprogram a computer to run a different program. So they're able to monkey around with them, for lack of a better word, in the laboratory in order to cajole them into behaving like embryonic stem cells. So they're not embryonic, no human life is taken. There are cells, but they are reprogrammed. They're induced to become pluripotent. The term pluripotent means they have the capacity, the potency, to become numerous different types of cells, or numerous different types of tissues also. So these are the 5 basic types. And we'll have questions at the end, so if you have some. Fetal stem cell research. No success. No treatment or clinical applications for disease or illness have been approved by the FDA. Embryonic stem cell research. No success. No treatment or clinical applications have been approved by the FDA. Cloned embryonic stem cell research. No success. No treatment or clinical applications for disease or illness have been approved by the FDA. To date, the FDA has not approved any of this research in human trials because tumors have developed in animals. These teratomas, that's what the tumors are called because they grow hair, flesh, and teeth and eyes. They're pretty nasty looking. These teratomas grow because embryonic stem cells multiply and divide at such a pace that you can't control them. So the scientists, they just can't control them, and so they're very dangerous because they're unpredictable and you can't stop their growth and you cannot cajole them like you can adult stem cells to turn into certain types of tissues.